Microbes are the fastest evolving organisms on this earth. As soon as new antibiotics are discovered, they start developing resistance to these drugs. Let us see how these tiny creatures develop antimicrobial resistance. The first mechanism is antibiotic modification or degradation. Bacterial proteins are synthesized in machinery called ribosomes. Antibiotics such as aminoglycosides bind to these ribosomes and inhibit their protein synthesis. Some bacteria produce enzymes called transferases which chemically modify the aminoglycoside antibiotics. An example is N-acetyltransferases, which add an acetyl group to an aminoglycoside antibiotic. This modification stops the antibiotic from binding to the ribosome and therefore it will be ineffective against the pathogen. Some bacteria produce hydrolytic enzymes that can degrade antibiotics. For example, enzymes called beta-lactamases inactivate beta-lactam antibiotics such as penicillins. In order to overcome this problem, strong beta-lactam antibiotics which are less susceptible to beta-lactamases were developed. However, the pathogens have evolved in their resistance by producing extended spectrum beta-lactamases or ESBLs which can degrade these strong beta-lactam antibiotics also. Some antibiotics are reserved as last resort drugs which are only prescribed when all other antibiotics fail to cure the infections. Unfortunately, some ESBLs are capable of degrading the last resort drugs as well. As a result, the bacteria are becoming multi-drug resistant or superbugs which are very difficult to kill. Modification of target or binding site. Most antibiotics work by specifically binding to their targets, thereby preventing the physiological function of these targets. Some bacteria develop resistance by modifying the antibiotic targets. An example is methicillin-resistant Staph aureus. Methicillin is a class of penicillin antibiotics that was commonly prescribed against staphylococcal infections. They bind to penicillin-binding proteins called PBPs in Staph aureus, which is encoded by a gene called MECK. Sometimes, Staph aureus acquires a mutation in the MECK gene, resulting in an altered PBP, which does not have an affinity towards methicillin. The antibiotics becomes ineffective and the MRSA continues to grow. MRSA is one of the leading causes of mortality or death. Reduced antibiotic concentration. Most antibiotics have intracellular targets such as ribosomes. Also, a known intracellular concentration of an antibiotic is required to exert its effect on a pathogen. Some bacteria develop mechanisms to prevent drug entry and reduce the accumulation of drugs inside the cell. Antibiotics enter a bacterial cell through protein channels in the membranes called porins. Bacteria express few or defective proteins so that antibiotics cannot enter the cell to reach their target. Another mechanism is through efflux pumps. Bacteria efflux or transport toxic compounds outside the cell through efflux pumps. They develop resistance by increased efflux of antibiotics, thereby reducing their concentration inside the cell. Biofilms. Bacterial communities produce a thick polysaccharide matrix called biofilms. Antibiotics cannot penetrate through this matrix and therefore is ineffective against the pathogen. So what are the consequences of antimicrobial resistance? We have not heard the term antimicrobial resistance any time before and nothing serious is happening around us, right? Then why should we worry for this antimicrobial resistance? If any of us or our relatives are admitted to the hospital, sometimes we have seen a delay in the discharge, right? The reason might be persistent fever, relapsing fever, or no response to the antibiotics. All these reasons are due to antimicrobial resistance. Due to the delay, our hospital bill also increases, right? In fact, many hospital deaths are occurring due to multi-drug resistant microbes. In other words, antimicrobial resistance leads to longer hospital stays, higher medical costs, costly and toxic alternatives, and increased mortality. According to a review on antimicrobial resistance, resistant infections caused around 7 lakh deaths worldwide in 2016 alone. If we are not careful, Antibiotic resistance will lead to 10 million deaths globally by 2050 and cost up to $100 trillion. Imagine if microbes develop resistance against all antibiotics available on this earth, an era may come where common infections and minor injuries can kill people, and also complex procedures such as surgery and chemotherapy become too risky. It is a natural phenomenon that microorganisms develop antibiotic resistance to survive on this earth. But it is in our hands to control this by proper usage of antimicrobials.